Hello guys, this is the first lecture in genomics. So I'm going to teach you the genomics subject for MSc2 biotechnology. Um, so genomics is the very big field actually. If we are talking in terms of genome or DNA or the complete sets of DNA or complete genome of the organism, it's a big thing, huge thing. As far as we know that we know the model of DNA, how it works, the phenomenon of central dogma of life, that is the DNA turns it into RNA and then ultimately it translates it into the protein. So this is the biggest thing what is happening in a single living organism, and that governs all the functions in the body of organism. So first of all, we'll see what is mean by this is an introductory lecture. So we'll see very little details about the genomics, and then we'll go ahead with the detailed topics. So genomics is an area within genetics that concern the sequencing and analysis of an organism's genome. So once we do the sequence of the genome, that means we are going to find out what is the sequence of that particular genes, and based on that sequence we come to know what kind of language, what kind of proteins can be built from that sequence. It involves the study of all the genes which is present in the DNA or mRNA and the up to the proteome level. Okay, so the term genomics is first coined by 1986 by Tom Roderick. Okay, he is the geneticist at the Jackson Laboratory in Maine. Okay, during the meet about the mapping of the human genome, he coined that whole term. So it is the study of all the genes present in an organism as far as we know. If we define the genomics, we can define in terms of like a discipline in the genetics that applies recombinant DNA, DNA sequencing methods, and bioinformatics to the sequence, assembled and analyze the structure and function of the genome. So this is the comprehensive definition of genomics, and you must learn this definition by heart. Okay, it includes the studies of intragenomic phenomena such as heterosis, epistasis, pleiotropy, and other interactions between loci and alleles within the genome. If you see the history of genomics, it actually eventually starts with Fred Sanger in early in 1970s, because the concept of genomics is laid by the Sanger, who first uh, sequenced the complete genome of virus and mitochondria. Later in 1972, the Walter Fiers and his research group, they became the first to sequence the gene, and they sequenced the gene of bacteriophage MS2. Then 1995, Hamilton Smith and his team became the first to sequence the genome of free living organism, that is Haemophilus influenza. Okay, that was the first full genome, a full genome of organism has been sequenced. And then before this 1995, in 1988 only, based on the Sanger sequencing method, the concept has been put forth before the different scientific society, which is funded by National Health Institute of these US and defense institution, also defense department, that we can map the whole human genome. And the research has been led by different health department of or CDC even though included or involved within it. If you see the difference in between genetics and genomics, there is a little difference. Like genetics is the study of heredity, right? It is the inheritance that we study, what genes inherited throughout the generation. That study we generally do in the genetics. Where in the genomics, it is the study of entirely of an organism's genes, which is present in that particular generations. The gene here in the genetics is referred to specific sequence of DNA which is present on a single chromosome at particular site. And here we refer genome refers to an organism with entire genetic makeup. Genetics involves the study of the function and the composition of a single gene, and here in the genomics we addresses all the genes and their interrelationship. Subfields of genomics. The different research areas of genomics can be as follows. Like first of all, structural genomics, functional genomics, and comparative genomics. See, the main goal of genomics is that the sequence the entire genome by cutting it into small, manageable pieces, that is fragments. Okay. Second, assemble the entire genome from pieces, that is the fragmentations we assembled, we arrange them in a sequential manner, and then we understand the expression of that genes. Why to sequence genome? This is the very important questions actually. Sequencing the genomes helps understand how the genome as a whole and how the genes works together to direct the growth, development, and maintenance of entire organism. The genome sequence will represent a valuable shortcuts, okay, thus helping to find out the genes much more easily and quickly. And this is how, what kind of proteins it provides, what kind of different systems actually it tells us, these all these things is actually answered by genome sequence, or the why to sequence this whole gene. Technically, we generally construct so many libraries while we doing the whole genome sequences. We can construct the genomic libraries, cDNA libraries. C means complementary DNA libraries. See, now what is complementary DNA libraries? We know that the genome contain intron and exon, where introns are non-expressible and exons are expressible units. When DNA transcribe into mRNA, then we know that the whole set of mRNA after the splicing mechanism, it is completely made up of a exon or expressible characters or expressible sequence. And based on that mRNA, if we construct the DNA material, that is complementary DNA, we can construct libraries of just expressible genes within the whole organism or from the whole genome, and that libraries we generally call cDNA libraries. Then the DNA hybridization, restriction enzyme mapping, DNA sequencing, PCR, these all the techniques actually is necessary, and we need to study them in detail. There, what actually we do in genome sequencing, like we break the genome into smaller fragments with the help of different restriction endonucleases. We sequence those smaller pieces. We construct these pieces of sequences of short fragments together, and then like there are after the preparation, after the constructing, we analyze where what different loopholes are there, what different spots or what do we call the different short stretches is remaining or not, that we can see based on these constructions. There are two types of approaches. One is hierarchical shotgun sequencing and the other one is whole genome shotgun sequencing. The hierarchical shotgun sequences is more best, and it is the it is the technique where we have sequenced whole human genome. Okay, useful for sequencing genome of higher vertebrates, okay, than the content repetitive sequences, and the whole genome shotgun sequences. So we use it for smaller sequences, smaller organism. HGP, Human Genome Project. We'll see in details, but this is just a short information. It is an international scientific research project with the goal of determining the sequence of chemical base pairs which makes up human DNA, and of identifying and mapping all the genes of human genome from both a physical and functional standpoint. The Human Genome Project was formally founded in 1990 by the US Department of Energy and the National Institute of Health. Okay, and it was declared completed on 14th April 2003. So the basic goal of this HGP were like to identify all the genes and their functions in the human DNA, to determine the sequences of 3 million base pairs to make up the human DNA. So There are different goals actually, like in the medicine field or medical field. We can understand what different inherited diseases are there, what kind of different inherited diseases are actually coming up through the genes or mutation, because of mutation. These all these information, the bunch of information actually we are getting from the sequencing. To develop the tools for data analysis, to obtain the physical map of human genome, and ultimately to store the information in the public databases so that the different laboratories, different research institutes can access that data for their own research. There are amounts of benefits and applications. It can be used in the field of medicine for early detection of genetic diseases and its diagnosis and treatment. It also useful in the field of agriculture. It is also like useful to study the evolution through different mutation lineages. Okay, and it is also helpful in forensic science. So this is the introduction of um, the genomics subject. I hope so you understood what we are going to study in step by step in the lecture. Okay, so thank you very much. Thank you.